Hello, good morning. Welcome to the 40th Structure Meets Function webinar organized by Instruct-ERIC. My name is Marjetka Podobnik. I'm from National Institute of Chemistry, Ljubljana, Slovenia. Let me just first introduce briefly Instruct-ERIC. So Instruct-ERIC is a pan-European network for structural biology. So it's a single point of access to technology and expertise in integrated structural biology. So Instruct-ERIC has 17 members, which are mainly countries, European countries, but also organizations such as EMBL, and also Israel is a member. Instruct also has several infrastructural centers where one can apply for research visits to those centers and use the high-end technology to do experiments in the field of structural biology. So you can also apply for internships, which can be from 3 months to half a year to do some work in these centers. And Instruct-ERIC also organizes workshops and additional training especially for PhD students and postdocs. And of course, every year one can also apply for R&D funding for like startup projects, also for younger researchers. So, but if you want to have more information get more information about Instruct-ERIC, feel free to go to this contacts listed here and to the webpage. And last but not least, we in Slovenia, for these members, for people from Slovenia, we also have an Instruct-ERIC branch in Slovenia, and we have our own webpage where you can see also our activities. We also organize workshops, and we also provide additional information. Or if you need more information on Instruct-ERIC, I'm a national coordinator, a scientific representative from Slovenia in Instruct-ERIC, and my email is here written in the upper right corner. And so with no further ado, I would like to introduce our first speaker, who is Nika Žibrat. So Nika studied food science and nutrition at the University of Ljubljana, and then she, for the master's degree, she moved to the Netherlands, to the Wageningen University, where she enrolled into medical biotechnology. And after that, she came back to Slovenia, where she is now at the National Institute of Chemistry at the Department of Molecular Biology and Nanobiotechnology. And she studies these proteins that are expressed by the pathogenic microorganisms and attack plant membranes. She uses different approaches, approaches to study the mechanism of action of these proteins, and including structural biology. So Nika also was, she also applied for internship at Grenoble ESRF last year, and she spent 3 months there. She probably would tell us something about it. But now I welcome Nika to present her work.
Thank you. Let me just share my presentation. Okay, is that visible? Yeah.
Yes.
Hi, welcome everyone. Thank you for joining in such big numbers. I heard that these are quite outstanding numbers for this kind of webinars. My name is Nika. I'm super happy to be here. And I will present, as Marjetka already mentioned, I will present something about NLPs, which are plant plasma membrane binding and pore forming toxins from distinct plant pathogens. So first, part of the reason why I'm presenting today is because Slovenia became an Instruct-ERIC member, I think, 2 years ago. And I was lucky enough that I was able to apply for this Instruct-ERIC funded internship and was accepted, and went to EMBL Grenoble for 3 months, where I worked at Marquez Lab. And the goal of my internship was to improve my skills in terms of sample preparation, crystallization, and computational methods of crystal structure solving. So I worked at the HTX lab, which is part of the Marquez Lab, and it's a high throughput crystallization facility, which offers high throughput crystallographic services. And this was of very very big value to me because the automatization that they have there, I have never seen before, and it really broadened my horizon. So I would recommend anyone who has the option to apply. If you have any off-the-record questions, just like practical, my email is also here, and I'm happy to help in any way that I can. So now on to the science. The rhizosphere and phyllosphere of terrestrial plants are home to a number of microorganisms, many of which are potentially pathogenic at certain stages during their association with plants. Over the course of evolution, pathogens have developed a plethora of effectors, such as proteins or small molecules, to manipulate the cellular processes of hosts and establish parasitic relationships with them. Many of the effectors encoded by plant pathogens remain understudied, limiting success in targeted disease prevention and loss of crop yields. And the losses of crop production due to pathogen-induced plant diseases average about 20% of the world's harvest, with a further 10% loss during post-harvest, and this severely limits the production, quality, and safety of the food today. We study NAP1-like proteins, or shorter called NLPs. These effectors constitute one of the largest microbial protein families, and they represent an important and severely understudied toxin group. They are expressed across many phyla, namely bacteria, oomycetes, and fungi, and more than 1,700 NLP genes have been identified in the genomes of more than 500 species until today. And the most prominent example of NLP encoding pathogens are Fusarium, Botrytis, Plasmopara viticola, and of course Phytophthora infestans, which was responsible for potato famine in 19th century in Ireland. Lipid membrane destruction by pore-forming toxins is a well-known mechanism to damage animal cells, but is less prominent and less researched in plants. And in contrast to bacterial or mammalian plasma membranes, the composition and structure of plant plasma membrane has only recently begun to be deciphered and exhibits extreme complexity. The lipid receptor for NLPs in plasma membrane is a glycosyl inositol phosphoryl ceramide, or shortly we call it GIPC. GIPC lipids are the most abundant plant sphingolipids and are assumed to be located almost exclusively in the outer leaflet of the plant plasma membrane. The structure of GIPC so far is poorly understood, particularly with respect to their sugar heads, which contain a variable number of hexoses or pentoses. And it can be from 2 to several these sugar moieties. Interestingly, GIPCs of monocots and dicots can bear the same type of terminal sugars, but in different numbers. Predominantly, what is thought today, 2 sugar heads in eudicots, which is called series A GIPCs, and 3 sugar heads in monocots, and this is called series B GIPCs. Binding studies and X-ray crystallography revealed that the cytotoxic NLP-PA from oomycete Phytum afanidermatum, which is far the most studied NLP today. So this NLP forms complexes with the terminal monomeric hexose unit of the GIPC, and the binding leads to several conformational changes, such as widening of the binding crevice and magnesium.
Can you show also your pointer? We don't see your pointer.
Yes.
If you just use the. Oh, now I see it. Okay, thank you.
Okay.
Sorry.
Okay, let me just move my Teams window so I can. Okay. Yes, so these are the terminal sugar heads that I was talking about. And this is when this complex was co-crystallized with the terminal sugar moiety. We can see that these loops are widening, and this crevice then accommodates this terminal sugar. And this is the magnesium ion, which the position in apostate is here, and the position of the magnesium ion changes to this position. And this was the sign that this was the first indication that this might be the binding place, and that this opening accommodates these GIPC sugar head groups. And there has been a long-standing hypothesis that NLPs are able to exhibit toxicity only towards dicot plants. And this was due to the fact that the insensitivity could be explained by the longer GIPC's head group, as you see here in this very simple scheme. So 3 sugar heads, terminal sugar heads for the series B from monocots, and NLP then is binds this GIPC in this crevice, and there is not enough proximity due to this additional terminal sugar head comparing to dicots, and the NLP cannot actually come into contact with plant plasma membrane and then induce NLP mediated damage. So now I will talk about our results. When I say we, I talk about our laboratory, not just my results, but from everyone who was and is part of this laboratory and contributed to these results. So we have established that plant plasma membrane lipid models, we have established plant plasma membrane lipid models that we are able to now incorporate into liposomes. I incorporate GIPCs into different lipid membrane systems. But first, I should say that GIPC lipids are not commercially available, and we do orovalone purifications and extractions of these lipids. We get them from series of homogenization, extraction, washing in order to obtain them, and it's time and cost strenuous process, and the yields are very limited. However, we have able to modify the existing protocols for certain assays to incorporate them into the liposomes, planar lipid membrane bilayers, and so on. And we are able to study NLP binding and pore formation on these liposomes. We perform sedimentation assay, as seen here, and surface plasmon resonance. With such liposomes, we infiltrate these NLPs into tobacco leaf apoplasts. And with this, we have been able to test the binding of two NLPs, one toxic and one non-toxic. So NLP-PIA, as already mentioned before, is very highly studied and it exerts toxic effects on tobacco upon infiltration, while this NLP from Hyaloperonospora aridopsitis does not exert necrosis and toxic effects on tobacco leaf apoplasts. And the binding of these both NLPs was tested to see whether binding correlates to the toxicity. And as seen here on the surface plasmon resonance and tobacco infiltration, indeed, this non-toxic NLP does not bind to the membranes consisting of GIPCs. The important part of the protein was supposed to be more or less flexible loops, L2, L3, and LC1, which dictates the widening of the crevice to accommodate GIPC sugar heads. After establishing GIPC is the receptor for toxic NLP-PIA, the aim was to characterize the clustering on the membrane, pore formation, and pore size. Transmission electron microscopy, seen here, and high-speed atomic force microscopy were performed to assess the shape of clustering or oligomers and the depth of the insertion of the protein into the membrane. The nature of the pores were assessed with the addition of NLP-PIA to planar lipid bilayers using orbit from nanion, and with giant unilamellar vesicles to which NLP-PIA was added together with fluorescently labeled dextrans of different sizes. The results showed that the protein does cluster on the membrane, that the pores are transient, as seen here from the opening and closing in terms of changes of electrical current. And the pores appear smaller than 10 kilodaltons in size. As you can see that 4 kilodalton dextran penetrated the membrane upon addition of the protein, while bigger dextrans did not. And the insertion of the protein to the membrane does not reach throughout the whole lipid layer, but only the upper leaflet. This is seen from here, but for more information, this is the paper where this was published, so feel free to read it if it's of interest to you. So the NLP-PIA is most research NLP, and it was almost exclusively studied for its mechanism of binding and pore formation to GIPC-rich artificial lipid systems. But what about other toxic NLPs from phylogenetically unrelated plant pathogens? Do they all work in similar fashion by binding to GIPCs? Do they cluster and induce leakage on the membrane? We don't know. And secondly, the only artificial membranes tested so far were from tobacco GIPCs, meaning from dicot plant. What about other host plants? What about monocots? So to address these questions, we used 2 NLPs from different phylogenetic origin, one oomycetal and one fungal. We used the microfluidic systems where we could selectively move GUVs into the diffusion chamber with the electronic tweezer and added protein solution to the system. The protein would reach these vesicles gently with diffusion, so we could observe real-time events on the liposomes under a confocal microscope. So here is a representative experiment with microfluidics. So we used NLPPA from Phytum aphanidermatum, for which we already know is spore forming on dicots, but it's not known if it's spore forming also on monocots. So here we can observe many things. First, we see that there's green labeled protein. We labeled NLPPA with Alexa. And here we can see giant unilamellar vesicles containing, in this case, tobacco GIPCs, in the lower case, leak GIPCs. And we have encapsulated them with a red Alexa, which is approximately 0.6 kilodaltons in size. And we have added the protein. Here I'm going to show you a video. So in this way, we could observe what happens when the protein reaches the membrane. It binds to the membrane. And at the same time, we see that this red Alexa dye is released from the vesicles, and the vesicles become black. We can also see that the vesicles lose their shape. They start to collapse. And this happens for a large majority of the vesicles that we tested. So this collapse, we think, might be due to the fact that the protein uses shear force to bind and to destabilize the membrane, and therefore the vesicles collapse due to probably also some mechanical forces. So here we can observe that this happens in tobacco and in leak containing membranes. So we get this graph by following all different fluorescent signals on this video. So we get the red dye signal from the inside of the vesicle, which then get emptied. We get the green signal on the membrane, which is the protein that binds, and we get this blue curve, which is the signal of the protein in the surroundings. So we can actually assess how much is the percentage of the protein that binds, and how much is just in the background. So then we also compared time binding and time release of the vesicles. So time binding, this means 50% of the protein, of the total amount of the protein that we saw in the chamber that was bound, and we compare them between tobacco and leak. And also time release. Time release was time when 90% of the dye was released from the vesicles, and we see that in tobacco and leak, in tobacco it happens a bit faster, but the difference is not, it's not that vast that we would take another look, more detailed look into it. So what we can see from this, that NLPPA does form pores on dicot and monocot model plant plasma membranes, which was previously thought that it's not possible. Okay, so then we wanted to test in the same way if this is true also for another NLP, which is of another phylogenetical group from fungi. So this fungus Monilioptera perniciosa attacks cacao plants and causes witch broom disease, which severely hampers cacao production. And it has 2 NLPs, and one of these NLPs is MPNAP2. So we have tested this MPNAP2 in the same way. We have labeled it with green Alexa. We have encapsulated red Alexa in the GUVs, and we repeated basically the same experiment. And on the video, we can see that also this protein binds to the membrane, and the leakage of this red Alexa dye occurs. However, this protein, although the effects in the end are the same, we can observe some differences. The protein seems to be less aggressive towards the vesicles, meaning that the vesicles do not collapse. Also, the timing of how fast the leakage happens is a bit prolonged compared to NLPPA. However, the effects in the end, in terms of pore formation, seem to be pretty similar. So this was the first time that was established that not only omicetal, but other NLPs can also form pores, not only on dicot, but also monocot plants. So now on to the structural part. As I said before, 1,700 NLPs, NLP genes were already identified, but only 3 crystal structures exist so far. So we decided to study another NLP from another oomycete, which has been functionally tested, but not structurally characterized yet. So we have decided to study it on with 2 different structural approaches. So this protein is NLP from Phytophthora parasitica, which is also a very renowned plant pathogen. It attacks, I think, more than 50 plants. And we have studied this NLP with NMR. So first, we have prepared isotopically labeled protein, as one does for NMR, and we assigned the backbone and side chain resonances. And what was immediately recognized was the inherent flexibility on the protein due to the many absent and weaker backbone signals, which can be attributed to the conformational change dynamics and the broadening of the signal. So on the left, the colored in yellow are the parts of the backbone which are exhibited such features, so this intermediate exchange. And on the right, we see the bundle of 20 best structures that was generated by a final round of refinement after NMR structure determination. We have a fantastic NMR department in our institute. So Peter will also present some of the work. But we have done this in collaboration with people from that department, from the NMR facility. And what we also observed is that NMR spectra and the NMR structure in solution clearly shows the intrinsic flexibility of the protein, which likely reflects the functional reason towards the adaptation to the interactic partners. So as it was already the hypothesis before that these loops are kind of the gateway to binding to GIPC. And the exchange broadening observed in several segments of the protein can be attributed to conformational exchange dynamics to micro and milliseconds timescale. And this means that the conformational ensemble is available for interacting partners, indicating conformational selection binding process. We have also solved the crystal structure of this same NLP, and upon inspection, we realized that the fold is pretty much conserved to the rest of the 3 structures already solved. But we have observed some differences in this region, the binding crevice. So we have used CAST-P to locate and measure the concave surface, which is the binding crevice. And we have immediately saw that the binding crevice in NLPPA, which we know binds well and is pore-forming and is toxic, is more than twice the size of NLP from Phytophthora parasitica. So we thought, okay, let's see how this correlates with the activity of this protein. Could this be the reason that the protein behaves differently? So we did some functional assays, and we observed indeed that in this functional assays that this protein acts a bit differently. Well, not a bit differently, but less potent, let's say. So we first did infiltration into tobacco leaf apoplasts, and we immediately saw that this NLP from Phytophthora parasitica, comparing to other two NLPs that I showed before that are toxic and pore-forming, stops being cytotoxic towards tobacco at much higher concentration. So first sign that it is less potent. Then another thing that we did was the sedimentation assay, where we used tobacco, leak GIPCs, and the control, which was without GIPCs. And what we saw is that the binding of this NLP from Phytophthora parasitica is much less pronounced comparing to other two proteins, which was both true for tobacco and for leak containing membrane, GIPCs containing membranes. And then we also did surface plasmon resonance to see to compare these two proteins. So we did this with small unilamellar vesicles containing GIPCs from tobacco, and we saw that with different concentrations, NLPPA shows not only higher response units, but also NLPPP has less pronounced this dissociation phase, meaning that the binding is less stable, or as I would maybe even call it, transient. But the binding is still there. So yes, this protein does act less potently in terms of binding and toxicity. So we went to investigate the structure a bit more. Oops, sorry. So we saw that after the inspection of this binding crevice and the loop surrounding it, that the intramolecular interactions between the side chains of amino acid residues on the beta strand here and on this small loop LC1 here contributes to rigidity alongside the general amino acid composition of loops, which can, of course, influence the rigidity of the whole area. And the molecular dynamics that was done in collaboration with people from Kemijski Inštitut in Italy confirmed our observation that NLP from Phytophthora parasitica indeed has much more rigid loops, L2, L3, LC1, comparing to NLPPA. And the dynamical properties also differ between these two proteins. And this is another indication that these loops indeed might be important for GIPC binding and the overall toxicity of the protein. So one pronounced difference that we saw here, there are some, but I will not go into all of them, is, for example, here, this histidine, which is here inside of the binding crevice, forms hydrogen bond with this glutamate, and this really fixes the structure, this binding crevice together. While in NLPPA, for example, which is toxic and pore-forming, this does not happen. So cases like this were observed, and this is worth investigating further with some mutational analysis and so on to fully grasp the importance of this area for GIPC binding. So in summary, the mechanism of NLP-mediated pore formation is not yet understood. The determinants of NEP1-like protein binding to GIPCs are largely determined by loops, which determine binding crevice capacity to accommodate the polar sugar heads. And microbial pathogens encoding NLPs are targeting different hosts, and it might be also that NLPs are more or less potent towards GIPCs originating from different plants. So more plant systems need to be tested. NLPs from diverse phylogenetic origin binds to GIPC in plant plasma membrane, cluster oligomerize, and form pores, respectively smaller than 10 kilodaltons. Most NLP structure this is the first NLP structure in solution, and it supports computational predictions of conserved and flexible regions that are vital for ligand accommodation. And yeah, I think I'm done. So I would just like to say thank you to Instruct-ERIC, to John for inviting me to present. to EMBL for hosting me, to our research agency, for Slovenian research agency for funding, my mentors Gregor Anderluh and Marjetka Podobnik, all of my colleagues, and all of you guys for listening. Thank you.
Thank you, Nika. So if there are any questions, please write them into Q&A section. So yeah, there is none yet, but maybe Nika, if so maybe into your first statement of the summary, when you say that the mechanism is not yet known, could you so which parts exactly? Because you mentioned that in that paper from Katja Piertz, some mechanism was already sort of suggested. So so which steps and do you think need further clarification? And yeah, so either binding or once it's bound, and then when and how then the membrane is disturbed, or what?
Yes, so I think that the most important thing is the characterization of the pore forming process, right? How it happens after it's bound. The protein is bound to the membrane. So there is no structural information about the pore yet. We don't know how it looks like. Also, it would be, for example, useful. These proteins are too small to be studied on cryo-electron microscopy, but it would be useful if we could get some insights into pore formation with cryo-electron microscopy, for example, on the liposome or on some nanodiscs, for example.
And when they maybe form like bigger, like oligomers, right? I mean, then you can see them, but like individual ones, yeah, they're small.
Yes, so far I know there have been attempts, but one of the big problems in this system is GIPCs, because we cannot get them commercially, meaning that also we cannot get them into as a homogenic sample, right? We have quite some unimpurities. Also, there are batch-to-batch differences when we extract, because there is no standardized process. So I think this is the main mishap that we have so far, because whenever we do try to characterize the pore, we do have cryo-electron microscopy in our department, but the samples seem to be quite heterogeneous, which then disables any further determination of the pore or assays like that. So yes, we do know that it binds to GIPCs. We don't know that it clusters on the membrane. We do know that there's some insertion, but not that deep. We assume that the pores are transient, so they're opening, closing. They're also, yeah, could be that there's some special, not so mainstream mechanism to this that we have still not figured out yet.
Okay, yeah, but the future is bright. So
yes.
There's a question from Milaya Stahl. How are GIPC levels and ratios look? How do GIPC levels and ratios look in leak? So what are the levels there? Same in all monocots?
How do the GIPC levels look like?
Because I guess the concentration of GIPCs in leak, I guess, and whether the concentrations of GIPCs are the same in all monocots. That's the question, I guess.
So I would not know how to completely answer what percentage of all lipids are GIPCs, because they are almost exclusively in plant plasma membrane. So there, I think they are around 40% of total lipids in plant plasma membrane are GIPCs, but I'm not 100% sure about that. But what I can answer is that GIPCs come in different forms, right? We say different series of GIPCs, meaning that they differ among themselves in terms of terminal sugar heads. And we have, this is unpublished data, but we have figured out that also which series is present influences the binding and the how pronounced the effect of the NLPs on the membrane is. So different different plants in general, not just monocots, have different series of GIPCs and different ratios. So for example, there is one paper from the Netherlands that even states that two different species of onion, two different cultivars of onion, can substantially differ in GIPC content. And that's why I said before, it's so important to test more than just a few lipid systems, because this can really influence how NLPs act on the membrane, the level of GIPCs of different series.
Okay, so I will just, we'll go to one question more and then we'll move on. So this is from Francesca Paoletti. She asks, how conserved are the loops involved in the binding? Can this be a reason for different mechanism?
So when it comes to amino acid composition, in our cases, when I compare, for example, this NLP from Phytophthora and NLP from Phytoma fanidermatum, we can observe differences in the amino acid composition, but also one of the loops is longer in NLP from Phytophthora. So we would need some mutational analysis to see if that matters, but there is more in NLPPP, there is more of this aromatic, bulky amino acids present in the loops, and this presumably makes them more rigid. So I would not actually know the number of amino acid sequence similarities, but they are not very conserved when it comes to sequence.
Okay, thank you, Nika, a lot for your talk, for a very nice talk. So in the interest of time, we have to move on. But if you have more questions for Nika, please just post them, and she can answer them later in the Q&A for you. So our next. Thanks again, Nika. And the next speaker is Dr. Peter Podbevšek. He is also from National Institute of Chemistry. Peter studied biochemistry at the University of Ljubljana, and then he did his PhD also. He enrolled in the program of biomedicine at the medical faculty, but then he did it actually at this institute. After the postdoc, after the PhD, he went to Trieste to CISA, which is the International School for Advanced Studies in Trieste. So basically, there he studied the structure of long non-coding RNAs, and actually he's been working on RNAs and DNAs throughout his career. On RNA and also non-canonical DNA and G-Quadruplexes. And also for potential therapeutic use, he studies the effect of chemically modified nucleotides and small molecules on DNA or RNA structure. Recently, Peter has been particularly interested in the effect of oxidative stress on DNA. And so this will be also a topic of his today's talk. So Peter, so you're welcome, and you for the start of your talk.
Yeah, thank you, Marjetka. Let me find the presentation. Okay, I hope this is on your screen.
Yes, it is. Thank you.
I'll just switch to pointer. Okay, time to switch gears. Like you said, we're spectroscopists. We deal mostly with NMR. So if this is a structure meets function webinar, we lean towards the structure end. And yes, usually we work with DNA and RNA, and today's talk will be on DNA. So as I'm sure most of you here are familiar with DNA base pairing, and usually G would pair with C and A would pair with T, and you would get a nice double helical structure, A or B type. But that's not always the case. Sometimes you get some funny structures, or let's call them unusual. And in specific cases, you can have G-G pairing, and you can have 4 Gs actually in a planar arrangement. As you can see here, also this face of G pairs with the Watson-Crick face of a different G. So this is what we call Hookstein base pairing. And these planar arrangements we call G-quartets. If you have more of them, and if they stack, we call that a G-Quadruplex, or a G4 for short. And you could have usually 2, 3, 4, sometimes 5 of these planes stacking on each other, and they are connected by some loops in different arrangements. So basically, G-force can be very polymorphic, as you will see later. So we have G-tracts, which are continuous runs of Gs connected by loops. So basically, you need a DNA or RNA sequence which has multiple Gs interrupted, preferably by non-G nucleotides, and then again, GGG something, GGG something, GGG. This is a very simplified sequence. So X doesn't always have to be X. You can have different number of Gs, and loops can be of different lengths. Can also include Gs, but let's keep it simple for this talk. So back in the day, this used to be a chemical curiosity. You would observe these structures in vitro in some diluted solutions, but then people started noticing this might have biological potential. So even in our own genomes, we would have highly enriched regions with G, so very G-rich regions. So one of these are telomeres, which protect the ends of our chromosomes. And in vertebrates, basically humans, you would have thousands of repeats of T2AG3. So TTAGGG, TTA, da, da, da. So, and yeah, I guess you can imagine that you can have lots of those G4 structures in telomeres. A different region topic of interest are gene promoters. Bioinformaticians find out that G-rich regions are also enriched in promoters of many genes. Of course, this G-rich sequences would be much shorter, so not thousands of repeats, but just a few, let's say, tens of guanines. There are reports that say that if you get G4 structures in promoters, this will either upregulate or downregulate expression of a protein. It's case specific. And as you saw in the title of my talk, and you have some indication here, ROS means reactive oxygen species, which are of some interest to me lately. The reason is simple. So we live on a planet which has oxygen in its atmosphere, and this also means that there are not just O2, but other reactive oxygen species. And of the 4 nucleobases present mostly in DNA, so A, G, C, and T, G would have the lowest redox potential, so it's the easiest to oxidize. And when that happens, you can get multiple products, but the major product is 8-oxoguanine. So if you look at position 8 here, you get oxidation, and also this N7 is now protonated, so basically you reverse hydrogen bond donors and acceptors on this position. And of course, if you remember the scheme of the G-quartet, this means something must go wrong, and it does. But normally, our cells can manage basal ROS levels. So you have enzymes that would remove specifically this nucleobase and fix it by inserting a regular G in its place. But if you have some spikes in ROS levels caused by all kinds of nice or not so nice things, these repair mechanisms are overloaded, and you can have persistent lesions in your DNA. So now let's go to the structure part. Some time ago, 5 or 6 years, with some friends in Japan from Kobe, they were looking at a G-rich region, short oligo, from the VEGF promoter. This is a known oncogene which harbors a G-rich sequence, which was well characterized. So vascular endothelial growth factor. We don't really care much about the biology, sorry. So this would form in a diluted solution a G-Quadruplex with a topology like this. So you see here loops are a bit different. They run on the sides, but more or less this G-quartet arrangement, these planes are common to all structures. This was characterized by a different group, let's say 10 years ago. And then they were wondering, so what happens if you incorporate an oxo-G into some position? So they did a number, but let's take this one, G4. So in all cases, the G4 structure would be disrupted. So basically you get a non-structured oligo. I promise these are the only NMR spectra here. So the way we identify what's the secondary structure of DNA in solution is that we see specific signals. I mean, we see signals in a very specific region of NMR spectra. And we can also differentiate AT, GC, or this G4 very easily. So G4s would give signals here around 11 ppm. And you can see if you have G3, G4, or G5 replaced by 8-oxo-G, there's basically nothing here. So DNA is non-structured. So how can we fix it? They were thinking, can we just introduce a short oligo which would slot into this G3 segment? And yes, you can. So this is a GGGTT short oligo, which can recover G4 structure, but it's not that easy. It needs a bit of help. So you have to add a nucleotide residue which has a conjugated pyrene. And this polyaromatic group would stack on this terminal G quartet and stabilize the whole thing. So if you just use GGG something, it doesn't work. You need this stabilizing group. And you can see if you add this thing to any of those mutated oligos, we get signals here and we're happy. So our G4 structure was recovered simply by adding a sort of fixer upper short oligo. But this is biologically not very relevant. Of course, this is more of a proof of principle. So later we started looking at, let's say, more relevant examples. If you remember the human telomere, I was talking about TTA-GGG repeats. And if you have a short oligo like this is again well known, will form a G-quadruplex structure. Again, some different arrangement of loops. They all look very similar in these schemes I have here, but you can imagine in 3D this can be very different. You'll see on the next slide. And this also, if you mutate most of the Gs, you lose the G-quadruplex structures. But some positions somehow tolerate this mutation very well. And one of those was this G21. So this position here, if you put in oxo-G, the same fold is obtained. Then on the other hand, if you replace or mutate G10 here, you again get a G-quadruplex structure, but a completely different fold. So the system somehow accommodates this mutation by rearranging, so topologically. And of course, this is thermal stability. You lose a bit in TM. So actually, you lose quite a bit here, 20 degrees. But it's still stable at of course, physiological temperatures, but these are in vitro conditions. So these should be quite stable. So what's happening? You see the normal G quartet, and then if we add an oxo-G, like I said, hydrogen bonds and donors rearrange here. So now hydrogen bonding needs to adapt slightly. but this is still a planar structure. And if you look at the, so oxygen here now in red, fits perfectly into the structure. This is planar. The backbone is basically unaffected. So yeah, all in all, it's all good in this specific position. So if we mutate this one, a G4 structure is lost. Then we started looking at a different system, a bit more diverse. This is from another oncogene, BCL2. Again, we're only interested in the structure. The interesting thing here is that this G tracts are of different lengths. So we have G3, G3, G5, and then G3 again. So here it's unclear which 3 Gs are actually involved in G-quartet formation. So again, if we look at the parent structure that was solved here, we see that some Gs are just excluded from the G-quadruplex core. So they're in the loop. And you get 3 G-quartet planes as expected. Then again, we mutate some Gs. Here I'm only showing 2 examples. We did many more. So if you mutate G19 here, which is in the G-quartet, what happens is it just pulls the whole strand. So register shifts. One strand up, and G19 is now, oxygen-19 is now in the loop. And now this G here, which is, sorry, G16, now falls into place of the lower G-quartet. The whole thing shifts by 1 nucleotide up. And if you replace G20, which is in the loop originally, this is more or less a silent mutation, so not much happens here. It all stays the same. This is also reflected in melting temperatures. You see we start with 76 here, 67 here. This silent mutation basically makes no change. And here we were surprised. We actually got a rise in stability with BCL-oxygen-19. If we look at the high resolution structure, which is, okay, a bit messy here. Let me see. Okay, I have a nicer representation here. So we see that oxygen-19 that is now in the loop actually forms a base triad with other loop nucleotides. So if we look at here, G19 in red, G6 in blue, and C21 in blue. So it contributes to a capping structure with its different hydrogen bonding arrangement, but actually brings the melting temperature up. But this is very unusual, so very case specific. So usually oxidative lesions are destabilizing. So when we had all these high-res structures, a few more that I didn't show, we were wondering, so what does this mean for DNA processivity? So normally enzymes are meant to work with double helical DNA. What happens if we have G4s in our DNA? Of course, many studies were done on this before, and generally they're considered as roadblocks. So for transcription, basically any enzyme that slides on the double helix, right? It can process Watson-Crick base pairs, but this can cause a problem. So we did a simple primer extension experiment. We incorporated our oligos with or without oxygen into a system like this. So a short template binding region with a primer, which is fluorescently labeled, a short linker, and then our oligo of interest. So the enzyme we just used the simplest we could find. The enzyme would try to replicate the template strand in the 3 to 5 prime direction, so backwards. And the way this works, it has to access nucleotides in sequence. So not like melting experiments where you melt the whole thing and kind of expect basically a two-state process, folded and unfolded. Here the enzyme has to work in sequence, so it has to access the first G here, and then the second, and so on and so on. So we mixed these reactions, and we actually took aliquots at different times, so we could monitor the reaction as a function of time. And we could analyze this by capillary electrophoresis, and we found which products are present at which time. So this is just an example, electropherograms. And you can see that we start with just the primer, and then slowly the primer is extended, and with time we get longer products, which is expected. And if we integrate this and make some fancy charts, we can see some data. Just a second. So we start with our well-known BCl2 structure. We also have 3D coordinates for this. So what we have here added is the linker. So the first 4 nucleotides here are the linker. And here we would have the template, the primer binding region and the primer bound. These are not shown. So primer extension starts here with the linker, A29, GDG. But as we go backwards, we will count with +1, +2, because we go in the 3 to 5 prime direction. So, and this is the population of products with time. So what we get is mostly +5 product, which is expected. So the enzyme works through the linker, and the first A, which is involved in some base pairing, but not in the G quartet, and stops before the first G, so it cannot replicate G24, or it's difficult to replicate, which is expected. But somehow it gets through it, and then there's also some +6, so it also has issues with +7. But once it gets through that, it kind of works its way up to +10. So the next G tract is again problematic. So this must be some triplex unfolding intermediate, which is quasi-stable. And then it just goes through the end to full length. If we have some mutations with oxo-G in the loop, so basically the silent mutation that I showed before, we get the same result. So these silent oxo-G can be bypassed easily if it's not structured. Now, if we look at more interesting examples where we have oxo-G with those in that base triad, we see again the first G is rate limiting, and even now this preceding nucleotide, for some reason, we're thinking because this loop is now a bit longer, there's a proximity issue with the enzyme, so it cannot put G24 in its active site. But more or less the result is the same. This G4 structure is hard to bypass. If we just take a control, so an oxo-G which destabilizes G4 at position 18 or whatever, we get a whole bunch of products at a lot of full length. So BCl2 is quite hard to work through. On the other hand, human telomeric structures that I also showed before are somewhat different. So again, in the non-mutated oligo, you see mostly enzyme is stuck with the first G or the second G, and then before the second G tract, so this, I guess, triplex unfolding intermediate, and we get some full-length product. If we have oxo-G in the G quartet, you see now we get lots of longer products. So this is highly destabilizing. We get lots of +11, so before this second G tract, +17 before the third G tract, so like a hairpin or something intermediate, and lots of full length. if we take this oxo-G10, which has the lowest TM, if you remember from before, this is the most stabilizing. So the enzyme basically just goes through, although this is present in solution, but it's highly destabilizing. I guess there are multiple reasons. One is that it's in the central G quartet, and one I'll show in the next slide. And if we do a control sequence, we get a messy profile, so a lot of long products. So that's our data. And what our hypothesis here is that, so if it really depends where your oxidative lesion is in the sequence. So the enzyme, this enzyme works by replicating in the 3' to 5' direction. If you have a mutation early on, it works through it and then is kind of stuck with unfolding intermediates, triplex or hairpin or whatever. If you have, but in this case, let's say this is oxo-G10, where you have a mutation late in the sequence, so towards the 5' end, the mutation or oxidative lesion is present in all unfolding intermediates and kind of is destabilizing all the way as the enzyme works through. So that's why we believe we get tons of full-length product. And so basically that's our data. Now, what's the take-home messages? I guess definitely that oxidative lesions affect how enzymes process G4 structures, and this must have an effect on all kinds of, binding of transcription factors, transcription itself, many things, which biologists should answer. So like I said, we're spectroscopists, we stick with spectra and let others do their part. So I'll stop here. I have to thank, so of course, our group, my supervisor, Janez. All those high-res structures were done by Stase, who is now in Barcelona. The first story with VGF was in collaboration with our friends from Kobe, Professor Sugimoto and Professor Takahashi. And I have to thank our agencies for giving us those few euros that we get. And thank you for your attention.
Thank you, Peter. Thank you very much for a very nice talk. So we have one question already by Alessia Levanova. So can these G4 structures be observed in RNA molecules? And if yes, what is known about their biological function?
Yes, thank you for that. Absolutely, yes. They are present in RNA as well. Specifically in RNA, they would only form a parallel structure. So all these G tracks would be parallel. So you always get those propeller tie loops, double chain reversal. Doesn't matter really. What's the function is a good question. Not so well known. So they were shown on short oligos, so basically short RNA segments that G4s form, also in RNA, in vitro. Segments taken out of messenger RNA and stuff like that. But the function is, yeah, there are some speculations, but honestly, it's not known.
So they were found in a coding region. I think
it's mostly in UTRs.
Okay.
But yeah, you can find them anywhere. Again, what the function is case specific. Some hypotheses are given, but on a very small sample. So I would refrain from giving any general answer.
Let's see any other. There are no questions left on the list. All these sequences that you have, this is all like mammalian sequences, or is like, can you say anything about conservation between different species, whether what sequences do you usually take from people?
Yeah. So the ones that I showed today are, yes, mostly from human genes or telomeres. They are found in basically all organisms. So
I guess in viruses as well, right? Viruses have.
Viruses. Let me think. That's a tough one. Okay, viruses, I'm not sure. That I would have to check, but bacteria, yes.
Okay.
I mean, yeah, so, okay, now bioinformatically, yes. Short oligos in solution, yes. Is this actually present in bacteria at some conditions, you know? So that's a different question then. So how to observe this in vivo is a bit more tricky. So there are some antibodies that will bind this. But then we get to the question, were G4s there before, or is the antibody inducing folding?
Oh, okay.
Yeah, but we believe that there are, now that we have quite a few studies that do show that G4s are present, at least in some part of the cell cycle, you know, not always, of course, because normally you also have the complementary strand, which would be C-rich in these regions, which also forms a secondary structure, which I did not touch today. And then there's also supercoiling and all that stuff. So, but yeah, the general kind of consensus now is that in some parts of the cell cycle, G4s are kind of not preferred, but let's say, yeah, the structural equilibrium shifts towards G4s.
Okay, thank you. Thanks a lot. If there are no further questions, I guess you can still, people leave it in the Q&A. But thanks again, Peter and Nika, again for your beautiful talks. And thank you all for attending. And then I guess now for the summer, the webinars are off, and then we start again in September. Thanks a lot and have a great summer. Okay, bye-bye.